cells that we extracted from our cheeks. The purpose was to illustrate and instill how DNA can replicate through PCR, the polymerase chain reaction. The technique consisted of extracting the cells in our cheeks, forming solutions with primers and polymerase, and then applying heat control. That's just like the general overview. The research questions were, can we replicate DNA by manipulating the PCR process? And then more of a side note, the secondary research question was, what was our genotype for the ALU gene? All right, in more vivid, thorough detail, the methods consist of extracting the cells using a salt-based mixture, swishing the mixture around in our mouths and then spitting it out, and that provided an easily accessible source of DNA that we could use from the cells. Then we used the centrifuges to bundle the cells, inside the little tubes, and then that provided an appropriate cell collection to use further on. We provided primers and polymerase to initiate the process, and then we used a thermocycle to manipulate the heat and then stimulate the replication thoroughly. And then lastly, we used gels to determine if the replication even took place at all. So that was like where we got our data. All right, now data. So we used a gel with DNA markings. We based this off the vibrance and the length of the markings to determine how much of the DNA was replicated and if we express that gene, whether homozygous or heterozygous. Let's see, for my group personally, we did have a lot of lanes, but unfortunately only 3 of them worked. But as you can see, the 3 that did work were vivid and were detailed, so we were able to assume that the replication did take place through the manipulation of the PCR process. And just as a little side note, based off my knowledge of the gel, the 3 lanes that did work are homozygous for the ALU gene. Errors. As with any experiment that we conduct, we have errors that occur that could harm our final results. So these errors could even be traced back to the procedures that are written for us to use collectively. Like, number 1, the experiment was done over an elongated constraint of time where we had to do it over a course of a couple of days, and throughout that time we had Kibler, who was placing the tubes in different spots, manipulating them on his own, so we weren't in constant contact with them. If we were able to work on this for a whole day, then we can knock out the experiment in a day, and then we could consistently be in contact, with the procedures in every step. So then that would ensure more valid data because we would just constantly be, hands-on. The second one would be the solutions that we use, like the measurements from the procedures, could have been too small overall. Since this is non-human errors, then I'm just going over like the procedures that could have affected the validity of our data. So if we had solutions that were too small to use, then that might explain why some of the lanes didn't work at all. All right, the conclusion. I conclude that DNA can be replicated through the acceleration of the PCR process, and I'm personally confident in this claim because other groups had markings in their lanes, but they weren't as vibrant, or they're a little bit scattered and a little bit confusing to read. But based on ours, we just see the 3 lanes that are vibrant, and they are very clear. For future research, the acceleration of the PCR process will give scientists further insight into the reproduction of life on a very microscopic scale. So we're starting from base 1, or like foundation, to understand how life replicates and just life in general. So thank you.